This article is titled, Acquired resistance of lung adenocarcinomas to gefitinib or erlotinib is associated with a second mutation in the EGFR kinase domain, and was authored by Pao William, Miller Vincent, Politi Katerina, Reilly Gregory, Samwar Ramel, Zakowski Maureen, Chris Mark, and Varmas Harold.
Lung adenocarcinomas from patients who respond to the tyrosine kinase inhibitors gefitinib or erlotinib usually harbor somatic gain-of-function mutations in exons encoding the kinase domain of the epidermal growth factor receptor, EGFR. We show that in 2 of 5 patients with acquired resistance to gefitinib or erlotinib, progressing tumors contain, in addition to a primary drug-sensitive mutation in EGFR, a secondary mutation in exon 20. Which leads to substitution of methionine for threonine at position 790 in the kinase domain. Tumor cells from a 6th patient with a drug-sensitive EGFR mutation whose tumor progressed on adjuvant gefitinib after complete resection also contained the T790M mutation. No tumors with acquired resistance had KRAS mutations, which have been associated with primary resistance to these drugs. Biochemical analyses of transfected cells and growth inhibition studies with lung cancer cell lines demonstrate that the T790M mutation confers resistance to EGFR mutants usually sensitive to either gefitinib or erlotinib. Interestingly, a mutation analogous to T790M has been observed in other kinases with acquired resistance to another kinase inhibitor, imatinib. In patients with tumors bearing gefitinib or erlotinib-sensitive EGFR mutations, resistant subclones containing an additional EGFR mutation emerge in the presence of drug.
We are article.tv. Links in the description below.